Organism consists of one or multiple cells, each engaging in metabolic processes to support life form. Within these metabolic reactions, enzymes and proteins play crucial roles in guiding molecules to undergo precise reactions. However, these complex structures are composed of smaller building units, known as amino acids. Surprisingly, there are only 20 types of these fundamental building blocks, and they are shared across different species. What if we could create a 21st amino acid or even more? Could we potentially create entirely new biological and chemical functions that cannot be achieved with the standard set of 20 amino acids? To begin, scientists were thinking how they could introduce a novel amino acid at the site of the start codon within the genetic sequence. To incorporate the new amino acid, tRNA molecules that carry the amino acid must possess the ability to recognize stop codons. And a new aminoacyl-tRNA synthetase, or synthetase for short, that is able to catch new amino acid must be created. The synthetase is an enzyme which primarily functions in capturing a specific amino acid from the cellular environment and attach the amino acid onto a specific tRNA. However, even though the ideas were achievable, it was hard to achieve back in 20th century. Challenge faced by scientists was to find a compatible pair of synthetase and tRNA molecule that could effectively cooperate. Another challenge was that introduced amino acids had toxic effects on organisms, potentially inhibit their growth. In 1998, Furter managed to introduce a novel amino acid into an organism. He achieved this by modifying the yeast tRNA and successfully incorporating the new amino acid into E. coli cells in response to the amber start codon. Amber start codon, identified by the genetic code UAG, represents one of the 3 stop codons in the genetic code. However, the success rate of Furter's experiment was not high enough to widely use. It wasn't until 2001 that Schultz and his team successfully developed the method of incorporating new amino acid into protein by improving Furter's experiment and successfully increased the success rate. Schultz's team used a tRNA which can recognize the amber start codon from a bacterium called Methanococcus genesis, or MJ for short. They introduced random mutations in 11 nucleotides on the tRNA to reduce the interference with E. coli's synthesis by eliminating tRNA variants that can be interfered. They proceeded to evaluate how efficiently other tRNA candidates recognized the amber start codon. This evaluation was conducted within E. coli cells. Plasmids carry an ampicillin-resistant gene, which included an amber start codon, were introduced into the cells. Along with the MJ tRNA candidates and MJ tyrosyl tRNA synthesis, or TRICE for short, the E. coli cells were then cultured in the presence of varying ampicillin concentrations. The best tRNA was then identified and shown as B in this diagram. It was later named as mut-RNA. The second objective was to enable TRICE to carry a novel amino acid, or methyl L-tyrosine. To enable TRICE recognizing the unnatural amino acid, 5 active sites of TRICE were replaced with adenine, which then dysfunctioned the synthetase. Those 5 substitute sites were then underwent randomly mutations, and were then tested the efficiency by placing them with the presence of chloramphenicol along with a chloramphenicol-resistant gene containing an amber start codon, mut-RNA, the new amino acid, and E. coli. If the cells survived, that means they had successfully abducted amino acids at the site of the amber start codon. To confirm they carried the unnatural amino acid, the team transferred the surviving cells to a new environment, which contained the antibiotic without the presence of unnatural amino acids. Since cells with the unnatural amino acid couldn't survive in this condition, their death in the presence of chloramphenicol indicated their possession of the unnatural amino acid. The mutated synthesis was then identified and named as mut-TRICE. It can be seen that 4 out of 5 active sites of original TRICE have undergone changes. Schultz's team pioneered genetic code expansion, or GCE, allows us to integrate new amino acids into proteins, and it has also become one of the most widely adopted methods in synthetic biology. As of 2023, the number of genetically incorporable unnatural amino acids has exceeded 200 types. Moreover, in 2020, researchers achieved another milestone by developing the first novel amino acid that organisms can synthesize autonomously, eliminating the need for a controlled experimental environment. This group also used GCE to address the challenge of cell permeability related to phosphorylated proteins by incorporating an unnatural amino acid, p-threonine. Incorporating unnatural amino acids into protein is no longer a mystery. What are the remarkable applications that the GCE can lead to? That's a story for another day.